Okay, here we are, last section of the chapter. Here in this section, we're going to be discussing what it takes to transport across big things, okay? Forget individual atoms like sodium and potassium. Forget small molecules like sugar. Here, we're looking for large, entire proteins, entire hormones, entire, well, cell debris. To do this, to get this across into or out of the cell, we're going to have to use what is referred to as bulk transport. Bulk, huge, moving big, huge things across the membrane. This is going to be, definitely this is going to be an active transport. And there's going to be a lot of energy expended to do this. We see two processes. Stuff moving into the cell, stuff moving out of the cell. Endocytosis. Endo means within the cell. Cytosis, the cell. Cyto. Anytime you see cyto, cell. Endocytosis is movement into the cell, whereas exo, out. Exocytosis, moving out of the cell. We're going to focus on endocytosis for this lecture. We see there are three types of endocytosis. Phagocytosis. Phago meaning eating, cytosis, cell. So it's literally a cell eating. Pinocytosis means small bits. Pinocytosis literally is the cell taking small little sips of the liquid around it to see what's there, what proteins, what compounds are there that maybe shouldn't be or should be there. And then finally, receptor mediated endocytosis. This is where something is able to bind to one of those glycolipid or glycoprotein receptors and trigger an uptake event. When you look at phagocytosis, you see what happens is a large particle here. That jelly bean looking thing is supposed to be like a bacteria. A bacteria comes into contact with one of your cells inside your body. What's going to happen is the plasma membrane is literally going to reach out, enfold around it, and pull it in. And it's now within a lipid bubble. That lipid bubble we call a vacuole. The vacuole will, fuse with, lysosomes. Remember, lysosomes are little small vesicles, small bubbles that are filled with digestive enzymes. So this way, that particle, whatever that jelly bean thing is supposed to be, will be broken down into individual amino acids, individual sugars, and things like that to be used by the cell. Pinocytosis, small little sips. Small little sips to see what's floating by. So this way the cell can sample the fluid around it. Remember, your cells are bathed in fluid. Whether it's your muscles, your liver cells, whatever, they're surrounded by fluid. Things are flowing by them and not just in the bloodstream. So as things flow by, cells take samples. What's there? It's kind of like when you, as you're walking around, you're sniffing to see what you smell. Well, this is their version of it. They're taking small little sips to see what's in their environment. And then finally, receptor-mediated endocytosis. What you see here is instead of randomly taking up stuff via pinocytosis or phagocytosis, whatever those stars are supposed to represent, will bind to a receptor. They are the signal that binds the receptor. It will trigger the membrane to fold in, surrounding it, and then putting it into a small vesicle. The vesicle which will then travel somewhere in the cell. The exocytosis, getting stuff that it's made inside the cell out, is the exact reverse of what we talked about with phagocytosis. Something is made, preliminarily made in the ER. It's trafficked via a vesicle to the Golgi. The Golgi does the final twists, tweaks, cuts, pastes, puts it together with other polypeptides if need be. And then that vesicle leaves the Golgi and goes to the plasma membrane, where it will then fuse. As it fuses, that vesicle membrane becomes part of the plasma membrane. What was inside the vesicle gets dumped out. Think about it. Base of your tongue, salivary glands. There are cells there in the salivary glands, in the goblet cells that make the mucus. They're also making digestive enzymes. You do not want the digestive enzymes free floating around in the cytoplasm. So they're going to be made in the ER, final twists, tweaks, cuts, paste, whatever in the Golgi, and then they're going to be trafficked to the plasma membrane of the goblet cells, where they're going to spew out into your saliva, into the gland, and out into your mouth the digestive enzymes.